Now let's begin our discussion of photosynthesis. You need to be able to name metabolic strategies based on energy and electron sources, just like we've been doing. To diagram the structure and function of a photosystem, as well as of a chloroplast in its entirety, explain the contrast of creating ATP and NADPH by light-dependent reactions, explain how electrons excitation provides that energy, and how the strength of chlorophyll A as an oxidizing agent to split water provides the electrons. Compare and contrast linear and cyclical electron flow. Track and count carbons as they're fixed during carbon fixation. Name important enzymes and molecules in the light-independent reactions. And then explain how the reduction of those fixed carbons yields an organic molecule with high free energy. That organic molecule with high free energy is going to be the same glucose that was used in the breakdown of cellular respiration. Now, in order for us to continue with our discussion in connecting these metabolic strategies, we need to define the types of organisms which conduct these different metabolic strategies. Auto means self, and so autotrophs are self-feeders. They obtain electrons from inorganic substances. They sustain themselves without eating anything derived from other organisms. So plants and algae are examples of this. They use the energy from the sun, photo meaning light, to be able to do anabolic reactions which help them to obtain these electrons. Heterotrophs obtain electrons from organic materials made by other organisms. These would include predators or decomposers. You'll note that these organisms are using materials which were created by autotrophs. Chemoheterotrophs obtain energy from the breakdown of organic molecules in the foods that they actually break down. For example, eating plants. In some way, directly or indirectly, photosynthesis actually nourishes the entire living world. Whether you're a bug eating the plant directly or a lizard which is eating the bug that ate the plant, ultimately it's these photoautotrophs that are actually going through and creating the organic compounds that all living things depend on. So tell me in Socrative, which of these reactions is an example of how a chemoheterotroph obtains energy for life? Good. Chemoheterotrophs use aerobic cellular respiration, the breakdown of glucose produced by photoautotrophs. So they don't actually create the molecule of glucose that was used. But since glucose is going to provide the electrons that are needed for a chemoautotroph or a chemoheterotroph, then they're going to be able to utilize those to get that energy. So photosynthetic plants contain endosymbiotic organelles called chloroplasts, and within them are thylakoid stacks that are critical structures because they do what? Well, thylakoids are going to have lots of membranes, a large surface area, which means they contain a large number of photosynthetic proteins, and all of that is going to increase the rate or effectiveness of photosynthesis. This is an example of where having a large surface area of membranes, of specialized membranes, increases their functionality. Now in Socrative, I want you to tell me the name of these structures. Go ahead and pause here so you can use this picture as reference and come back when you're done. Hopefully you recognize the outer and inner membrane, that these thylakoids are the names for the membranous sacs inside, and that they're surrounded by a liquid known as the stroma. When we take a look at photosynthesis, this is a complex series of reactions summarized as the following, where energy, carbon dioxide, and water are the reagents that are the reactants of this compound, or of this reaction, and that these become converted into glucose and oxygen. If you were to flip this arrow, you would see the exact chemical reaction which summarizes cellular respiration. So photosynthesis and cellular respiration in a lot of ways are opposite of each other, where carbon dioxide becomes reduced into glucose and water becomes oxidized into oxygen. If you were to take a look at our cellular respiration process, you would see that glucose becomes oxidized to carbon dioxide and that oxygen becomes reduced into water. Photosynthesis is going to be an endergonic process, as where cellular respiration was exergonic. Now let's take a look at a brief overview of how this process works. Light and water are going to feed into light reactions, where NADP+, which is an electron carrier molecule utilized in photosynthesis, and ADP are used to create ATP and NADPH. Light is going to be the source of energy. So this is an example of a phototroph, right? Photo refers to light. Since water is the source of electrons, and water is an inorganic molecule, you'll note there's no carbon, this is an autotroph, where autotrophs utilize electrons from inorganic sources. So the energy from light and the electrons from water are used to reduce molecules of ATP and NADPH. The byproduct which is produced is oxygen, and it's a waste product for the plant. So these light reactions, which reduce NAD+ to NADPH and generate ATP by a process known as photophosphorylation. In the stroma are light-independent reactions. This dark cycle occurs and ATP is going to be hydrolyzed, NADPH is going to be oxidized, and this recreates molecules of ADP and NADP+ that can be used by light reactions. These result in the production of sugars, which can be used in cellular structures such as the creation of cellulose or the storages of starch, or they can be broken down by cellular respiration in the plant. Plants don't just have chloroplasts. They're not just making sugar. They're also breaking down that sugar for energy. Now let's take a deeper look at these light reactions. In order for plants to harvest light, they need to be able to somehow transfer the energy from that light into a more usable form. Plants do this using pigments. So pigments can be excited by light, the energy from light. And when they do so, the electrons which are inside of them become excited to a high free energy state. When these electrons become excited, as with all molecules, they would prefer to fall back down to the more stable, lower free energy state. But this light energy is not going to be destroyed in order for this electron to be able to come back to a more stable state. And so it simply becomes transferred to another molecule. So from one pigment to the next, we get this energy transfer, where the light energy excites the electrons in the pigment. That pigment's electron goes to a high free energy state, and when it naturally falls back down to the more stable low free energy state, that energy is transferred to a neighboring pigment. It's important to note the electron itself was not donated away. It's only the energy from the pigment that can be donated. Photosynthetic pigments thus absorb light energy. Chlorophyll A is an extremely important and key light capturing pigment. I'm about to show you in a minute. It is the only one that can donate an excited electron. All other pigments, which are going to be found on the exterior of these systems, they simply transfer energy. So if we take a look at a photosystem, these are going to be protein complexes which are embedded into thylakoid membranes. When we talk about photosynthetic membranes and functional membranes, we're talking about the functionality of proteins in those membranes. For photosynthesis, we're specifically talking about the photosystems. So photon here represents light and the energy from that light, which is actually going to hit a pigment. These pigments, which are embedded into light harvesting complexes on the exterior of this photosystem, this pigment is going to be excited. And so that electron in the pigment is going to go to a high energy state. And when that electron falls back down to ground state, a more stable form, it transfers its energy to a neighboring pigment, which thus becomes excited and can thus then pass its energy on to its neighbor and neighbor and neighbor. Light harvesting complexes are responsible for the transfer of this energy from light to that core system known as the reaction center. So hopefully it doesn't surprise you that in the reaction center, a reaction occurs from here in the center of the photosystem. That ever important reaction is when chlorophyll A, a very special pair of pigments, donates an electron to a primary electron acceptor. So there are two types of photosystems. Both work in the same way that I showed you. So light harvesting complexes on the outside are excited by the energy from a light beam. Those pigments become excited and transfer their energy to a core reaction center, and then that reaction center is used to donate electrons away. But the two types of photosystems will do different things with the electron once it's donated to the primary electron acceptor. In photosystem 2, discovered second but acting first, the photosystem 2's chlorophyll can actually split water to replenish the electrons that were donated away. Photosystem 1, on the other hand, is capable of donating the electron to a molecule of NADP+, creating a molecule of NADPH. So if chlorophyll A has donated its electrons and been oxidized, it's now given that away. It's oxidized. It has no more electrons to give, unless it can replenish its electrons. In order to do so, chlorophyll A is actually going to break a molecule of water. This is why chlorophyll A is known as an oxidizing agent. It is going to oxidize water into oxygen gas and two hydrogens from which it can steal their electrons, thus oxidizing them. That oxygen gas is a waste product, and this is why plants give off oxygen. It's not to help you breathe. Plants give off oxygen because they stole electrons from water to reduce a pair of chlorophyll A molecules so that they can repeat this reaction. Once those electrons are donated to a primary electron acceptor in photosystem 2, they're given to an electron transport chain, which pumps protons and donates those electrons later down the system. This electron transport chain creates a proton motive force, just like you've seen before. And there's ATP synthase in these thylakoid membranes, which can be used to create ATP. The only difference here is that protons are pumped into the thylakoid space, which is different because in cellular respiration, we pumped protons into the intermembrane space. But since this isn't a mitochondria, we have different spaces in the cell. Those electrons, once they've gone through the electron transport chain, are donated to chlorophyll A. So the terminal electron acceptor in this case is actually another photosystem. Those electrons in chlorophyll A get excited and re-energized by these light harvesting complexes, re-harvesting energy from the sun, and then that electron pair again gets donated to a primary electron acceptor. But in this case, photosystem 1 is going to donate those electrons to a pair of NADP+, reducing it to NADPH, and going off to do other jobs in the cell. So photosystem 1 replenished its electrons from photosystem 2 and the electron transport chain. So when the electrons were given away to the primary electron acceptor and eventually NADPH, in order to replenish new ones, it fed off the electron transport chain. But photosystem 2 doesn't have an electron transport chain that feeds it. And so in order to regenerate those electrons, it needs to split water. Only photosystem 2 can split water. The process by which we created ATP here is known as photophosphorylation. It's this electron transport chain passing electrons from the primary electron acceptor to the terminal electron acceptor, which is photosystem 1, and along the way pumping protons to create a proton motive force. That proton motive force turned the rotor of ATP synthase and catalyzed the production of ATP. But since this process is ultimately driven by the energy necessary in the form of light to keep the system going, we call this photophosphorylation. Now, the system that I just showed you is known as linear electron flow. It involves two photosystems, both photosystem 2 and photosystem 1. It requires water to be split to replenish the electrons, and it produces both ATP and NADPH, because if you can replenish electrons, you can also give them away at the end. Sometimes, however, water just is not available, or in a very limited sense. And so the cell is going to utilize electrons over and over and over again, not giving them away to NADPH, but actually feeding them back into the electron transport chain. Photosystem 1 can do this all by itself, feeding the electron transport chain with the same pair of electrons that it accepts at the end. And then it will energize those electrons using the light from the sun before donating them back to the electron transport chain. And in this way, the cell can actually produce more ATP.